Neuroscience is a field of research that has been a rapidly developing field of science over the past 20 years. One particular topic has been of high interest: locating and understanding the specific cells that represent particular memories, also known as Engram cells. Engrams are neurons that experience memory-dependent changes in expression and excitability. Simply put, creating new memories physically changes neurons' composition, structure, and function. Today, we are going to be discussing changes that were observed in Engram cells within the dentate gyrus, a small section of the brain located in the hippocampus whose cells are involved in memory recall. A paper done by Michele Pignatelli et al., published in 2019, will be our primary focus, more specifically figures 2 and 3. First, we are going to look at the researcher's experiment, which aimed to study fear memory through changes in the behavior of mice over several days and in multiple environments. On day 1, the mice were fear conditioned in environment A, meaning they were shown a specific cue, or in this case a visual cue, like light, that was followed by a shock. This occurred all throughout day 1 until the mice were conditioned to freeze upon seeing the light cue in apprehension of the shock. At night they were removed from environment A, but on day 2 the mice were reintroduced to the same environment and shown the same visual cue again. However, this time there was no shock paired with the light stimulus. After a set amount of time passes, the mice were introduced to a completely new environment, environment AB. Now, environment AB is different from environment A in all ways except for the presence of the visual cue that preceded the shock on the first day. What is interesting is the behavior observed after the light cue in environment AB. Previously on day 1, the mice learned to freeze after the visual cue was presented. During the recall in environment A on day 2, they again froze despite no shock following the cue. Interestingly, when mice were moved to environment AB, some of the mice froze and some of them didn't. So what was the differentiating factor? The amount of time that elapsed before being introduced to environment AB after A. So when mice were introduced to AB only 5 minutes after the recall in environment A, they did not freeze after the visual cue. However, mice that were introduced 3 hours after the recall did freeze. Here we have the data supporting the research, and when looking at day 2 test in AB, we see a significantly reduced level of freezing in the 5-minute group, but not in the 3-hour group. The researchers found this behavior very interesting and wanted to know what was causing the behavior changes. Why were some mice freezing and some not? So when they went into some neurons located in the dentate gyrus, again an area of the brain associated with memory recall, and looked at ion channel expression in the membranes, they found that an inward rectifier potassium channel, known as KIR 2.1, had time-dependent alterations of expression. Now that sounds really complicated, so let's define what an inward rectifier potassium channel is. Essentially, it's a pathway or channel through the membrane of the neuron that allows potassium to flow out of the neuron and keep it at its natural state. So, when the expression of KIR 2.1 is changed, the neuron's ability to reach its natural state will also change. In the mouse's case, 5 minutes after the recall in environment A, when they are introduced to environment AB, the KIR 2.1 channel is down-regulated. This is not observed in the 3-hour group. When KIR 2.1 is down-regulated, the neuron becomes hyper-excitable, meaning it fires much easier than it normally does. You can see this in the image at 5 minutes. The neurons in the dentate gyrus indicated by the white arrows are darker than at the 3-hour mark. This is because KIR 2.1 is stained using a red dye in this image. Another way to say this is that the neurons at the 5-minute mark are less red, meaning that KIR 2.1 is down-regulated. In summary, at the 5-minute mark, the neuron is more hyperexcitable due to the downregulation, but at the 3-hour mark it is not hyperexcitable. So what does the hyperexcitability of a neuron have to do with a mouse freezing or not freezing after the light show? To answer that question, we have to analyze the pairing of hyperexcitability and freezing. The 5-minute mice do not exhibit freezing in response to the lights and have a hyperexcitable dentate gyrus neuronal engram, while the 3-hour mice do freeze and do not have hyperexcitable neuronal engrams. The researchers hypothesized that the overexcitability of these neurons in the 5-minute group causes the mice to more easily remember the recall of day 2, where no shock was applied after the light. That is to say that the 5-minute mice remember that no shock will be administered after the light because their neurons are hyperexcitable, and correspondingly have their KIR 2.1 channels down-regulated. While this study is incredibly fascinating and has many advanced applications, there is still a lot left to be understood, and there are some limitations. Our hypotheses, based upon their results, is that there are two competing memory circuits. One circuit based out of fear, causing the mouse to freeze, and the other based out of safety, stopping the mouse from freezing. To test this, we thought that mapping neuronal circuits connected to the dentate gyrus would reveal the answer.